So Shayla, mahalo for all of your work, and let's get started here.
Great.
So Shayla Waiki today will be presenting assessing the connectivity of on-site sewage disposal systems to nearby shorelines and water quality in Keaukaha, Hawaii, USA. Let's give her a warm welcome.
All right. Thank you, Dr. Colbert, for that introduction. That cuts down what I will be saying on my introduction here. But I would like to start off by saying that for the past 2.5 years, I have been working on my thesis research that focuses on sewage pollution right here in my hometown of Hilo, Hawaii. I would have never imagined that I'd had the chance to conduct research in the waters that built the person I am today, and let alone lend a hand in trying to solve pressing issues like sewage pollution here in Hawaii. This project definitely holds a special place in my journey through science and academia. Okay, let's hope this works. Okay. All right, so I'd like to start off by discussing the importance of coastal waters worldwide. Coastal waters play an important role in the livelihood of many coastal communities, which is especially present here in the Hawaiian Islands. As many of you know, these waters serve as areas of recreation or gathering places for large portions of the local community. In addition, coastal waters also serve a key role in sustenance living and providing food for families, much of which is harvested in nearshore waters. Here in Hawaii, coastal waters are also used in cultural practices, ranging from teaching the art of throwing net, fishing, free diving, and even ceremonial practices called hi'ubai. To many, myself included, the ocean has a cleansing nature, one that may be used for emotional and even physical healing. However, water quality in coastal waters are affected by sewage pollution. The waters that once provided its cleansing and healing capabilities are threatened by sewage that enters the environment. Sewage contains inorganic nutrients, pathogens, endocrine disruptors, and many other toxins that is being introduced into these waters. The presence of these contaminants in coastal waters can have impacts on both ecosystem and human health. Sewage pollution in coastal environments can be detrimental to the organisms and ecosystems that are present in the area. Some of these impacts include harmful algal blooms, decreases in oxygen within the water column, and increases in microbial abundance. Excess nutrients in the environment can also alter benthic communities, causing phase shifts, changing from what once was a coral-dominated community to non-algal-dominated community, as seen in this picture here. The presence of sewage in waters that are frequently recreated by humans can affect human health. A study by Chauvel, 2003, reported that swimming and recreating in sewage-polluted waters results in approximately 120 million gastrointestinal diseases and 50 million cases of severe respiratory diseases. In addition, the consumption of filter-feeding organisms, like oysters, harvested from sewage-polluted waters, have caused 4 million cases of hepatitis A and E and 40,000 cases of chronic disability like liver damage. This goes to show that the health of coastal waters also determines the health of coastal communities, who are heavily reliant upon these resources. So basically, there are two main categories of sewage that can enter the environment: treated and untreated sewage. On-site sewage disposal systems are often used in rural areas to dispose of sewage. There are 3 different types of OSDS: septic tanks, aerobic treatment units, or ATUs, and cesspools. Septic tanks and ATUs provide little treatment to wastewater before being released into the environment, while cesspools provide no treatment at all. This figure here on the bottom provides a basic schematic of how cesspools work. Cesspools are simply a hole in the ground, as depicted here, that collects sewage from infrastructures. The sewage contaminants represented by these brown and green dots are introduced into the environment through pores and fractures of rocks. These contaminants sooner or later reach the groundwater. The flow of groundwater transports the OSD contaminants to nearby shorelines through submarine groundwater discharge, as seen here. Areas with high densities of OSDs located in close vicinity to the shoreline is susceptible to sewage pollution. Within the state of Hawaii, there are approximately 110,000 OSDs, 49,000 of which are classified as cesspools and located right here on Hawaii Island, as seen in this figure here. One area on Hawaii Island that has high densities of OSDs in close vicinity to the shoreline is the Hilo Bay region, which is right over here. The Hilo Bay region has approximately 8,700 cesspools in use. So in addition to OSDs, municipal wastewater systems and sewage outfalls are another mechanism of sewage disposal in urban areas. Municipal wastewater systems collect sewage from households, businesses, and other infrastructures via sewer pipes and is responsible for treating wastewater before releasing it back into the environment. In this schematic here provided, it shows the basic, the basic mechanism of treatment for wastewater. The wastewater is collected from homes and other infrastructures that then enters into the sewage treatment plant. It first goes through a primary treatment phase that removes solids from the wastewater. And then depending on the facility type, wastewater can also go through a second round of secondary treatment. Treated effluent is then released into sewage outfalls, where it is then carried to the site of discharge, often disposed of in the ocean. So here in Hilo, Hawaii, the Hilo wastewater treatment plant is Hilo's only wastewater facility. I will refer to the Hilo wastewater treatment plant as HWTP. HWTP is a secondary treatment facility which uses chlorination as a form of treatment. After chlorination, the treated effluent is released into the sewer pipes, where it travels to one last pumping station onshore, and then into the sewage outfall, where that extends 1.3 kilometers offshore, where treated effluent is discharged. So the use of smell in science is still a relatively new field, partially due to the fact that being able to measure smell scientifically is difficult. However, the sense of smell is important to us as humans, and researchers have identified that we are able to discriminate between at least 1 trillion olfactory stimuli, which is much more than we are able to discriminate using other senses. The sense of smell and being able to pick up on the presence of odors in certain areas is oftentimes one of the first things we notice. The presence of odors and how we interpret them allows us to describe places and also shapes these experiences of these places. Odors that we encounter in coastal regions range from fishy, sulfury, and salty. Well, that is just some of the few that I have encountered. I'm pretty sure that there is much more that one could list. One specific odor, the smell of sewage, is usually distinct and often coincides with the presence of sewage or other foul contaminants. The use of smell in science should be further developed because it can inform important information about certain areas. All right, so this study was conducted in Hawaii along the Keaukaha coastline, which is right over here, that is located within the Ahupua'a of Waiakea and the Moku of Hilo. The Keaukaha coastline ranges from rugged basalt rock to black sand and a few white sand pocket beaches. Keaukaha sits on the southern flank of Mauna Loa volcano, an area made up of sub-area basalt lava flows, and consists of numerous freshwater springs that feed into the coastline. Keaukaha is also an area susceptible to sewage pollution. HWTP is located just southeast of the Keaukaha community, right around here. And also the HWTP sewage outfall pipe extends 1.3 kilometers off of a popular beach called Puhibe, indicated by this red star. In addition, many homes within Keaukaha, Hawaii still utilize OSDs for sewage disposal. So the objectives of this research was one, to identify the connectivity of OSDS to nearshore waters through the use of dye tracer tests. The second objective was to characterize water quality from HWTP inlet to outfall. Third, compare water quality of OSDS impacted springs to HWTP outfall plume. And lastly, to document the intensity and extent of the smell encountered in Keaukaha, Hawaiʻi, and identify any environmental factors that are associated with the presence of smell. All right, so in order to assess both water quality of OSDS impacted springs and HWTP, water samples were collected and analyzed for sewage indicators. Water samples were analyzed for fecal indicator bacteria, what I will refer to as FIB, and three specifically, Enterococcus species, Clostridium perfringens, and Bacteroides species. Water samples were also analyzed for delta-15N and delta-18O and nitrate, as well as nutrients. The nutrients analyzed for in this study consist of nitrate and nitrite, phosphate, silica, ammonium, TDP, DOC, and TDN. So in order to determine the connectivity of OSDS to nearshore waters, dye tracer tests were conducted using a non-toxic and organic dye called fluorescein. Fluorescein was added to OSDS over a 10-hour period, and for this study, we conducted dye tracer tests on 3 cesspools and 1 septic tank. In order to determine if dye was present at the shoreline, 12 to 13 shoreline springs were monitored for dye at low tide for up to 2 weeks after dye addition. At springs where dye was observed, water samples were collected and analyzed for sewage indicators. To assess the water quality of HWTP from inlet to outfall, influent, effluent, and pre-outfall samples were obtained from HWTP. In addition, the sewage plume produced by HWTP outfall pipe were also sampled. The end of the outfall was accessed via boat, and a CTD was used to determine the thickness of the outfall plume. Water samples were collected in a Niskin bottle at depths of 0 to 3 meters. These water samples were then analyzed for sewage indicators. Then lastly, to document the intensity and extent of the smell encountered in Keaukaha, Hawaiʻi, we created a Google Form survey called the Pilau Meter. The Pilau Meter recorded community observations of the presence and intensity of smell encountered in Keaukaha, Hawaiʻi. The survey consisted of 13 questions that ranged from date, time, and location, from questions that asked the type and severity of smell, ranging on a scale from 1 to 5. Other questions consisted of environmental conditions and demographics. So for this survey, the types of smells were categorized into 3 different categories: good or normal, fishy, and sewage. Correlations between Pilau Meter responses and environmental variables were carried out to identify if there are any associations between the presence of smell and any environmental factors. The documentation of this situation was one component the Keaukaha community requested within our research. So now as I move on into the results from the study. So dye was observed at the shoreline from all 4 dye tracer tests conducted. The dye at the shoreline were first initially observed in low concentrations that were then followed by a quick increase in dye concentrations, reaching its highest dye concentration shortly after. This quick increase was then followed by a slow decrease in dye concentration over the following days. If we take a look at this graph here, this represents one dye tracer test that we conducted in Keaukaha, Hawaiʻi. On the x-axis, we have date, and on the y-axis, we have fluorescein measured in parts per billion, or ppb. This red arrow indicates when dye was added to the OSDS, and the green circles indicate a spring with dye presence, while the yellow circles represent a background station. So we can see here on October 16th, the green circles identify low concentrations of fluorescein observed at the shoreline. But on October 17th, there was a rapid increase in dye concentrations, reaching its maximum value of approximately 15.2 ppb. Over the following days, dye concentrations decreased slowly. The dye tracer test done in this study identified a travel time of 20 hours to 3 days for sewage contaminants from OSDS to reach the nearby shorelines. So these dye tracer tests we conducted in this study have identified that sewage contaminants are moving at flow rates of approximately 130 to 213 meters per day. These flow rates that we have identified are faster than previously measured and modeled elsewhere in Hawaii. So another study in Keaukaha used dye tracer tests to determine the connectivity of septic tanks, ATUs, and cesspools to nearby water bodies. This study identifies flow rates of 3 to 137 meters per day. The fastest flow rate identified here is equivalent to the slowest flow rate identified within Keaukaha. Another study done in Lahaina, Maui, used dye tracer tests to determine the hydrological connections between a wastewater treatment plant injection well and coastal waters. Flow rates from this study were determined to be 1.7 to 2.1 meters per day for sewage to travel from the site of injection to coastal waters. This again is much slower than the flow rates of both the Keaukaha study as well as the Keaukaha study. In addition to measured flow rates, a modeling study done by the Hawaii Department of Health used a 2-year travel time to estimate OSDS that pose the greatest risk to nutrient loading in coastal environments. In Hilo, the 2-year time of travel was set at approximately 6 to 8 kilometers inland from the shoreline, which is depicted here by this purple line. The 2-year time of travel means that sewage contaminants from OSDS located 6 to 8 kilometers inland would take 2 years to reach the Hilo shorelines. Using the modeled travel time, sewage contaminants will be moving at a flow rate of approximately 11 meters per day, which is still much slower than we have identified in Keaukaha. However, if we were to use the average flow rates measured in Keaukaha of approximately 169.8 meters per day, it would take 47 days for sewage contaminants from sewage systems located 8 kilometers inland to reach the shoreline, which is much faster than the 2-year travel time. Fast travel times for sewage to reach the shoreline is concerning because it reduces the time of which biological processes are able to treat wastewater and rid them of harmful bacteria before entering coastal waters that are heavily recreated. So now in regards to water quality of HWTP, both FIBs, Enterococcus species, and Clostridium perfringens were significantly higher in influent samples compared to effluent samples. However, after the treated effluent was released from the wastewater treatment plant and made its way to the sewage outfall, FIB levels increased. Pre-outfall samples were higher in FIB concentrations compared to effluent samples, as seen in these graphs here. The graph on the left depicts Enterococcus, while the graph on the right depicts Clostridium perfringens. If we take a look at the graph on the left, on the x-axis, we have the 3 different wastewater treatment stages. And on the y-axis, we have Enterococcus concentrations measured in mpm per 100 milliliters. So the trend that I just explained is present here. Influent samples are significantly higher in Enterococcus concentrations compared to effluent. And then from effluent to pre-outfall, we see an increase in Enterococcus concentrations again. That same trend can also be seen for Clostridium perfringens. So in addition to Enterococcus and Clostridium perfringens, Bacteroides species was also detected in all stages of wastewater treatment. The decrease in FIB from influent to effluent suggests that chlorination treatment being used by the HWTP is effective at reducing FIB. However, treated effluent is being transformed upon leaving the wastewater treatment plant and arriving at the sewage outfall. Next, let's take a look at dissolved organic carbon, or DOC. DOC significantly decreased from influent to effluent samples and continued to decrease in pre-outfall samples, as seen in this graph here. On the x-axis, we have the 3 different stages of wastewater treatment, and on the y-axis, we have concentrations of DOC measured in micromoles per liter. DOC is the only nutrient that decreased among the different stages of wastewater treatment. All other nutrients increased. All right, so now we take a look at the nutrients present in different wastewater treatment stages. Nitrate and nitrite, phosphate, and TDP significantly increased from influent to effluent samples. and even then continue to increase from effluent to pre-outfall samples. The graphs here on the top, it depicts nitrate and nitrite, and on the bottom is phosphate. So if we take a closer look at the graph on the top, on the x-axis, we have the 3 different stages of wastewater treatment, and on the y-axis, we have concentrations of nitrate and nitrite measured in micromoles per liter. So it is also key to note that silica, ammonium, TDN, delta 15N, and delta 18O and nitrate were not significantly different among wastewater treatment stages. In addition, the nutrients measured in HWTP effluent were lower than effluent from Kihei and Lahaina, Maui. So while nutrients still remain in the treated effluent, this raises concerns because these nutrients are being introduced to waters that treated effluent are being discharged into. While the HWTP is a secondary treatment facility, there are other wastewater treatment mechanisms that targets nutrient removal. Such mechanisms include biological nutrient removal. In these systems, it is responsible for removing total nitrogen and total phosphorus for wastewater using microorganisms under different environmental conditions. The use of both treatment processes, chlorination for FIB reduction and biological nutrient removal, could serve as a better treatment mechanism to ensure pathogens and excess nutrients aren't reaching coastal waters. So finally, we make our way to HWTP outfall pipe. While sampling the outfall plume, we were able to see the surface oil produced by the discharge of sewage from the diffusers on the seafloor. Our study also found that surface waters near the end of HWTP outfall pipe were fresher and warmer than waters at a greater depth. So this data here is taken from one of our outfall sampling days, July 21st, 2021. The graph on the left depicts salinity, while the graph on the right depicts temperature. So if we take a look at this graph here on the left, on the x-axis, we have salinity, and on the y-axis, we have depth measured in meters, with the greatest depths here on the bottom. So here we can see that in the surface waters, until about 2.5 meters, there was lower salinity and fresher waters. Then if we take a look at the graph on the right, this depicts temperature on the x-axis in degrees Celsius and depth on the y-axis in meters. So here we see a trend where surface waters are much warmer than waters at a greater depth. Waters at the surface were about 25-ish, 25.2, 25.3 degrees Celsius. And this is indicative that we were actually measuring or collecting waters from the sewage outfall and not groundwater from shoreline springs, due to the fact that the average temperature of shoreline springs was approximately 19.1 degrees Celsius. The waters that we were sampling were in the 25 degrees Celsius range. So when comparing water quality at both the dye tracer shoreline springs and HWTP sewage plume, dye tracer stations had higher concentrations of Enterococcus species, nitrate and nitrite, phosphate, silica, TDP, and TDN compared to the sewage plume. These graphs here on the left-hand side depict Enterococcus, and on the right-hand side depicts nitrate and nitrite. So if we take a look at the graph on the left, on the x-axis, we have our 2 different samples. So the dye tracer stations and HWTP sewage plume. And on the y-axis, we have our Enterococcus concentrations measured in ppm per 100 milliliters. So here we see that that trend is present. In our dye tracer stations, there was higher concentrations of Enterococcus as opposed to HWTP sewage plume. And that trend can be seen for all nutrients listed here on this slide. So in addition to that, dye tracer stations had lower concentrations of Clostridium perfringens, ammonium, and DOC compared to HWTP sewage plume. So these graphs here on the left-hand side depict Clostridium perfringens, and on the right-hand side, DOC. If we take a look at the graph on the left-hand side, on the x-axis, we have our two different samplings, one being dye tracer stations and the other HWTP sewage plume. So here we see that in our dye tracer stations, there is very low concentrations of Clostridium perfringens, and the concentrations of Clostridium present in HWTP sewage plume were significantly higher than that of the dye tracer stations. All right, so the low concentrations of FIB within the sewage plume produced by HWTP outfall pipe could be due to rapid initial dilution. This rapid initial dilution can be a result of the momentum of discharge exiting the diffusers or the buoyancy difference of discharge effluent and seawater. In a field experiment in Brazil, they reported sewage plume dilutions of 35:1 in stratified conditions and 100:1 in unstratified conditions. With our study here, we identified a dilution factor of 35:1, with 35 parts seawater to 1 part sewage. So lastly, the results from our Hilo meter. The Hilo meter recorded a total of 85 responses. Of the 85 responses, 65% reported a good smell. These good smells were correlated with warm air and warm water temperatures. In addition, 17% of those 85 responses reported a fishy smell. Fishy smells were positively correlated with tidal height and negatively correlated with Wailuku River gauge height and water temperature in Hilo Harbor. It is to note, though, that these correlations were not significant. In addition, the 13% of the responses from the Hilo meter recorded a sewage smell. These sewage smells were correlated with wave peak period. And I would just like to note that we had wave activity data for a limited amount of time, as the Hilo wave buoy was out of the water for a certain time period. So these correlations observed for fishy smell with tidal height, it makes most sense, because as there are lower tides, more regions of the coastal area is exposed and could be contributing to the fishy smell. In addition, the negative correlation with Wailuku River gauge height suggests that on days with lower river inputs, there is a higher chance of the presence of a fishy smell. So, however, even though we were not able to identify much correlations between the presence of sewage smells and environmental factors, there were 2 instances where sewage smells were reported on the Hilo meter that also corresponded with HDOH advisories. The first incident took place in this time frame from June 17th, 2021 to June 21st, 2021. These are the dates where sewage smells were reported on the Hilo meter. And a couple of days later, on June 24th, 2021, the Hawaii Department of Health put out an advisory for a high bacterial count within Puhi Bay. So although the source of the high bacterial counts were not identified, the Hilo meter was still able to pick up on this incident. Next, the second incident where sewage smells were reported were in this time frame of January 29th, 2022 to February 22nd, 2022. Prior to this, Hawaii Department of Health put out an advisory on January 28th, 2022 that reported to avoid areas of Puhi Bay and the sewage outfall as wastewaters were discharged due to a power outage. So in conclusion, this study confirmed dye presence at the shoreline from all 4 dye tracer tests completed in Keaukaha. Sewage contaminants were identified to reach the shoreline in 20 hours to 3 days. In addition, water quality at dye tracer stations were different than that of the outfall plume. This study also identified that treated effluent is transformed once leaving the wastewater treatment plant and arriving at sewage outfall. Last but not least, although significant correlations with environmental factors and the presence of sewage smell was not determined, the Hilo meter was still able to pick up incidents where sewage or high bacteria counts were present that coincided with HDOH advisories. So with that, I would like to acknowledge everyone that's listed here on this slide. If it weren't for your guys' support, motivation, and dedication, none of this would have been made possible. And I would like to send out a special thank you to our project funders, Sea Grant and Ha'oli Maoloa Foundation, for allowing us to conduct and do these kinds of research here at home in Hawaii, especially in Hilo. And with that, I would like to mahalo each and every one of you for being here today. And I would also like to send out a special thank you to the homeowners who participated in our dye tracer research. And I can take any questions at this time.